hug. And all right, everybody's doing that. Thank you very much. That's very important. We have again today a fantastic speaker, scientist, who came from very far, and he's in person, which is wonderful. And again, it is a Friday, so we're celebrating. What do we celebrating, Johnson? No, we celebrate vision research. And with that, let's ask our wonderful friend to do introduction.
Hello, good morning, everyone. My name is Jiashong, a postdoc from Dr. Research Staff. Today, it's my honor to introduce Dr. Nikolai Artemyev. Today is a special day. You know, today is Valentine's Day. I hope everyone have a lovely and also wonderful Valentine's Day. And at such a special day, we also have a special speaker, Dr. Nikolai Artemyev, a leading expert in visual transduction. Dr. Artemyev is a distinguished professor in the Department of Molecular Physiology and also Biophysics, and then the Department of Ophthalmology and Visual Sciences at the University of Iowa. With over 30 years of experiences, he has made groundbreaking contributions to our understanding of visual transduction, particularly the molecular mechanisms regulating the photoreceptor function and the visual disorders. His research has been instrumental in advancing knowledge in key phototransduction proteins, including the visual G protein, transducing, and PD6, critical components for retinal signaling cascade. Dr. Artemyev earned his PhD in biochemistry in the Institute of Evolutionary Physiology and Biochemistry at the Russian Academy of Sciences. He later pursued postdoc research at the University of Illinois at Chicago, where he deepened his expertise in physiology and biophysics. His research focused on the visual transduction in rod and cone photoreceptors and molecular mechanisms underlying visual disorders. His lab employs cutting-edge structural and biophysical techniques such as X-ray crystallography, cryo-EM, small-angle X-ray scattering, etc. Dr. Artemyev's work has provided critical insights into structure and function of key phototransduction proteins, leading to many major discoveries, which he will share with us in today's seminar. Beyond his research, Dr. Artemyev is an active leader in the vision science community, serving as a reviewer for NIH study sections and international research foundations. His contributions have earned him numerous honors, including the American Heart Association's established Investigator Award. Through his pioneering studies, Dr. Artemyev continues to shape the field of vision neuroscience, offering new insights to retinal diseases and potential therapeutic strategies for vision disorders. Please join me in welcoming Dr. Nikolai Artemyev, who will present his talk in this special day, Maturation, Structure, and Dynamics of a Human Cone PD6. Thank you.
Thank you very much for this really kind introduction. I'd like to thank Chris for this invitation. I'm really pleased and happy to be here because I know that this is now a center of excellence for basic research in vision and also in translational research. So it's really a pleasure for me to be here. And yesterday I enjoyed a snowstorm. Yesterday I enjoyed rare rain here. And overall, this visit is just going great. So today I'm going to talk about maturation, structure, and dynamics of phosphodiesterase 6. And I would like to first thank two people, primarily Sneha and Dirich in my lab, who contributed the most to the data that I will be presenting today. So now, rods and cones employ homologous signal transduction cascades And the effector enzyme in these cascades is cyclic GMP phosphodiesterase. Right here, this is rod phosphodiesterase. It's a heterotrimer of alpha and beta subunits, also called A and B, PD6A and B subunits, and each associated with a rod-specific gamma subunit. And when these gamma subunits are displayed by activated transducer, the enzyme hydrolyzes cyclic GMP, and the channels get cyclic GMP gated channels get closed, and we have downstream signaling. Now, con employ con-phosphodiesterase, which is a homodimer of two PD6C subunits, and each bound to the con-specific gamma subunits. And phosphodiesterase are essential not only for phototransduction, but also for the health of photoreceptor cells. Because if there is a deficit in PDE6, cyclic GMP level rise, and the larger fraction of cyclic GMP gated channel becomes open, and influx of calcium, excessive, actually leads to apoptosis of photoreceptor cells. So mutations in ROD-PDE, they cause retinitis pigmentosa, and mutations in con-PDE, they result in achromatopsia. There is another way, besides mutations in PDE genes, to get deficits in PDE6, and that is PDE6 before that's okay. So PDE6. Okay, so PDE6, can you see this? So PDE6, before traveling to the outer segment for the transduction, needs to be assembled, it's synthesized in the inner segment, where it needs to be maturated, assembled, and then trafficked to the outer segment. And defects in maturation of PDE6 also in deficits of PDE6, and they result in even more severe retina disease called Leber congenital amaurosis type 4. And it turns out that for maturation, PDE6 rely on a specialized cochaperone APL1. APL1 is called so because it's homologous. Yeah, maybe this one.
Wir alle haben zu viele Sachen, die wir nicht wirklich brauchen.
Yeah, okay. Yeah. So anyway, so they require specialized chaperone APL. What is my answer? I'll just advance slides. So they rely on specialized cochaperone called APL1, and it's called so because it's homologous to aryl hydrocarbon receptor interacting protein called APE. They have similar structure and domain organization, and this protein was first identified to be linked to Leber congenital amaurosis, but then knockout of APL1 in mouse resulted in markedly reduced level of phosphodiesterase protein, no phosphodiesterase activity, and this led to severe retinal degeneration, and possibly it was determined that PDE is likely to be misassembled. So that led to suggestion that APL1 is a chaperone for phosphodiesterase 6, but direct evidence for that was lacking. So, and this one. Yeah, so now direct evidence for that was lacking. And we actually, several years ago, number of years ago, established first expression of phosphodiesterase heterologous expression in HEC293 cells. And what we found that there is no activity if you try to express phosphodiesterase itself. If you co-express with APL1, you get low activity, but small fraction of the enzyme is functional. But if you co-express enzyme with APL1 and PDE gamma subunit, and then of course you need to delete gamma subunit to release activity, you get very robust activity of phosphodiesterase 6, which you can see on this panel. So, and because the scale is different, its activation versus APL alone is about 100-fold. So that actually triggers further interest in my lab to continue to study APL1. And we solved subsequently crystal structures of the FKBP domain of APL1, shown in green, and the TPR domain, shown in blue. And FKBP domain of APL1, as we have shown, binds lipid modifications of PDE6, while TPR domains of FKBP containing co-chaperones are known to interact with the C-terminus of HSP90. And so subsequently, we hypothesized that HSP90 may be involved in maturation of phosphodiesterase 6. And we have demonstrated that the complex of HSP90 and APL1 is really critical for the maturation of PDE6. What we did is we use in cell culture, using this expression system, in cell culture, we've shown that inhibitor of the ATPase site of HSP90 attenuates expression of PDE6. And secondly, this expression is disrupted of functional enzyme when we mutate residues that are predicted to block this interaction. And specifically, we mutate these contacts with HSP90 contacts on APL, and PD expression was abolished. So then, a quick introduction about HSP90. HSP90 is a ubiquitous chaperones that are thought to work late in protein folding and activation, unlike HSP70 or HSP40 proteins. They don't just chaperone every unfolded protein. They late in chaperoning proteins, they induce active conformations of the protein, they help protein assembly, they stimulate ligand binding by proteins. And they don't chaperone every protein, they chaperone a subset of important signaling molecules, some of them listed in this table, including transcription factors, kinases, and many other proteins. And these proteins are called HSP90 clients. And how HSP90 chaperones these proteins has two subunits. It's homodimer, dimerized at the C-terminals, STD. There is NTD, N-terminal domain, in HSP90, and that N-terminal domain binds ATP and hydrolyzes ATP. And MD domain, middle domain of HSP90 protein is primarily involved in interaction with the client protein. So this APO HSP90 is open conformation protein. And when it binds ATP, it binds clients simultaneously and begins to close. And it closes in such a way that a segment of a protein becomes unfolded and threaded through the lumen of HSP90. And in a cycle of ATP hydrolysis, when ATP is hydrolyzed, it's first assumed closed state, threads the client, ATP is hydrolyzed, client is released, and then it's released in active conformation. So to further study maturation of PDE6, we decided to develop a more robust expression system where we can purify various components. And we established expression of PDE6 in SF9 cells. And this is what happened. And what we did, it's very simple. We infected cells with combinations of viruses, express a GFP fused phosphodiesterase, bind it to a GFP nanobody resin, cleave that from the resin using 3C protease, and then run, typically it's already clean enough, and we run size exclusion chromatography. And when we express PDE alone, we see a very, very broad elution profile. And also we saw 70 kilodalton protein always co-migrating with PDE6C. And we found that this protein by mass spec is HSP70. And there is also smaller protein of 40 kilodalton, which we believe is 40, but we just didn't do the mass spec on that. So, and it's very broad and aggregating protein. Our conclusion, PDE6C by itself is just aggregating protein. Now, next, we co-express PDE6C with APL1. And what we see is, you see that peak, small peak of protein, that peak corresponds to PDE6 protein. And we have activity now. For the first time, we have activity in the system, but the peak activity is shifted from the peak of the protein. So, and we analyzed and normalized the mouse retina extract by Western blot. We estimated that in this system, we obtain roughly a fraction of about 10% of active functional PDE6C, and it has same Km and Ki for inhibition by P gamma, but it's just a low fraction of this, but it's fully functional. So what's happened if we do co-express all three proteins, PDE6C, APL, and P gamma? Now we have a nice peak corresponding to approximately 250 kilodalton. That peak coincides with the peak activity, and by normalizing with rod mouse, rod PDE activity, we found that our con-PDE activity here, all protein is fully active, all. There is no unfolded protein. It's fully mature enzyme. So now, why phosphodiesterase 6 aggregates in the absence of APL1? We hypothesize, and I think actually it's not a hypothesis, it's probably, we can assume, it's a reasonable assumption that individual domains of PDE6 fold in the absence of APL1. They fold because we know there is a structure of GAF-A, GAF-B domain dimer of con-PDE expressed in bacteria in the absence of APL. So GAF domains fold, and most likely by the action of HSP70 and HSP40. So catalytic domains of phosphodiesterase 6, we fail to express that one. There might be multiple reasons for that, including that when we tried to do that, there was no yet beautiful structure from Chris of rod PDE. So our designs were not ideal. But anyway, so I think it's a reasonable assumption. So then why it's aggregating? And that's from the structure, from the Chris structure. So it's catalytic dimer, one subunit is shown as a surface. So we will just remove that subunit and rotate to see the dimerization interface, and red showing hydrophobics. And you can see that dimeric interface in PDE6 occludes, there is a lot of hydrophobic residues. So it's reasonable to assume that these guys, if they don't dimerize correctly, they will just aggregate. So, and also What that means is HSP-APL system works on monomeric undimerized yet subunits. And that's what's happening with homologous chaperone of AHR, aryl hydrocarbon receptor. So there is a structure in yellow, there is AHR plus B domain, APE in magenta, and there is HSP antidimer. Dimer. And as you can see, pass domain is loaded on HSP90, and fragment is threaded through the lumen. And APE acts as the cradle. It supports AHR when it's in the complex, in the maturation complex with HSP90. So that's one possibility. The other possibility is actually flowing from the structure of guanylyl cyclase C, which is actually somewhat homologous to retinal guanylyl cyclase. And I'm not ruling out that HSP90 system is working in supporting some of the maturation of retinal guanylyl cyclase. But here what's happening is that dimeric interface between kinase homology domain and dimerization domain is partially unfolded, and HSP90 together with another co-chaperone, CDC37, folds partially unfolded guanylyl cyclase. So that's the two options. So, okay, we thought, okay, if monomers are dimerized, we thought, let's do molecular dynamics. Let's do molecular dynamics of monomeric PD subunits, which we grab from Cris structure. And you can see immediate and very fast consequence of molecular dynamics is a kink in the linker region between GAF-B domain and the catalytic domain on both subunits. So from that, actually, yeah. So our conclusion from that is basically, yeah, so it may be that HSP90 and APL need to unkink that LH2 helix linking the GAF-B and the catalytic domains. But also, okay, so that's what HSP and APL do. What does gamma? At this point, we don't know for sure, but we think we know at what point gamma works on the HSP90 APL complex. So what we did is we expressed PDE6 without APL and gamma as usual, and bound to resin, EGFP resin. But this time we didn't elude that, but used that inactive PDE as a substrate to our chaperone component. So if we add to PDE, if we add H stands for HSP90, A for APL, PHA, if we mix together, we get some activity. If we mix all together with gamma, we have elevation of activity. If we incubate PDE, HSP, APL for some time, and then add gamma, we actually don't have any elevation of activity. What it means that PD got loaded on HSP90, APL did it work, but something happened. It's assumed conformation or get released, and gamma doesn't have any effect. But again, if we incubate PD, HSP, APL, gamma, after that, following that incubation, we incubate with a mixture. Again, we get elevation of activity. So we think that gamma acts in concert with APL at the later stage, and it acts on HSP90 PDE6 complex. And this is our current scheme. So we think nascent PDE domains are individually folded by HSP40 and HSP70, kinked or partially dimerized PDE6 are loaded on with the assistance of HOP HSP90 organizing protein HSP70 and HSP90 loaded on HSP90. And then in the absence of APOL and gamma, everything is misassembled and aggregates. If you add APOL, you get through transition complex, you get dimer protein that we see on gel filtration. It's a dimeric protein, but it's largely misassembled. Small portion of that protein actually does assemble correctly. But if you do it in the presence of gamma, that maturation complex actually releases fully assembled PDE. How that complex may look like. So we know how it looks. Oh, we tried that structurally analyze this maturation complex. But of course, first we decided, okay, let's go easy way. Let's try first to solve complex of HSP90 with APL1. We get cryo-EM structure, but the interesting thing, we found that if KBP domain is actually occupies, we don't see much of the TPR domain, but we were able to model the TPR domain at the bottom using cross-linking mass spectrometry. But APOL is actually, APOL of KBP domain is occupying client space, which clearly tells us that this structure is not very useful, because most likely what's happening when client is present, FKBP domain gets displayed, moved into proper position. But there are two complexes that are available now that include HSP90 and co-chaperones that are containing FKBP and TPR domain. One of that is, I already shown you, is AHR, APE HSP90 complex. And the second one is HSP90, GR, and FKBP52. And both complexes showing like ligand binding domain for ligand binding domains, and then the hinge region on linker region are threaded the lumen, and other domains such as they're not visible because they're flexible. Passé domain for HR and DNA binding domain for glucocorticoid receptor GER, they're on the other side. So what will happen if you submit PDE sequences? And that's what I did, submit monomeric PDE subunit, HSP90, and APOL to AlphaFold modeling. And out of 5 models that AlphaFold 3 typically spits out, 4 showing PDE, GAV domains actually not looking towards the lumen, but actually missing the lumen. But one model is actually turns out to be sensible, and I think it's consistent with everything that we know from literature and from our own experiments and molecular dynamic simulation. In that model, you actually can see that catalytic domain is cradled by APE, APL1, and the region is threaded through the lumen with the GAV domain on this side. So I think this is a good working model. I don't think this captures the exact interaction site, but it is closer to what we expect. So now summary for maturation. APOL and P-gamma, PDE fails to dimerize and aggregates. With APOL, PDE is able to dimerize, but it dimerizes in incorrect way. So it's misassembled. With small portion is assembled, but not in the rod. For rods, actually, without APOL, without gamma, only APOL, there is no activity at all. And we know that, we don't know why. But that's an interesting question for future research. But when both are present, APOL and PD gamma, they direct proper assembly of PD6. Now, an APOL and P gamma act on the catalytic subunit, apparently threaded through the lumina, which is P90. Likely somewhere near the connecting helix between GAP B and the catalytic domain. Okay, now going next, structure. Why study the structure of PD6? Because already there is a beautiful structure of rod PDE, and the homology suggests you will get a very, very similar structure. But there is a reason. Of course, we know that cones and rods, they do have very different responses. And cone responses are less sensitive and much quicker in kinetics. And that differences in physiology of roded cones is very, very important. And PDE6 was attributed as one of contributing factors. And historically, we know that continuous noise from classical study by Ricky and Baylor, that continuous noise is generated by spontaneous activation of PD6. And that sets the kinetics of photo response. And then at Pew and colleagues, they have shown that increase in spontaneous activity and steady state activity is the primary mechanism underlying shortening time to peak and decrease in sensitivity. And finally, we, a number of years ago, also generated a mouse model where we replaced rod PD with cone PD. Gamma subunit was still rod gamma. But then we still were able to see increased basal PD activity, faster responses, and reduced sensitivity. So that set up our rationale for studying cone PD. And we did it using our new expression system. So we expressed PD6 with APOL and P gamma, and we use both cone specific and rod specific P gamma. And the reason we did that, because we wanted to understand if these subunits contributing to the rod cone differences. So this is purification by mass photometry, really nice peak. This is for the data set with rod subunit. These are 2D classes, various frontal side, full size particles, different sorts of 2D classes. And again, just to mention, CGMP was added because it's known that CGMP binding to non-catalytic sites helps to stabilize, at least in cone PD, cone gap A domains. So now, this is just quick overview of our data processing. But what I want to point out is, since we have a large data set, by 3D classification, we were able to isolate subset of subunit that had ponytail. The others don't have. And ponytail, as we know from Chris' study, is a very interesting feature in rod PD. And what is exciting is it's also asymmetrical in cone PD, although cone PD is a homodimer. So, and we got the structure of this structure to about 3.1 angstrom. So now this is the structure, and let's put it for a spin, and you can see densities for 2 gamma subunits colored in green, and on the other side. So we see densities for all domains, GAF A, GAF B, catalytic domains, gamma on both sides, and gamma interacting with GAF B domains, and at the bottom, it blocks the catalytic site. So the ponytail and the GAP-A are more flexible, as suggested by local map resolution. And GAP-B and catalytic domains are fairly more structured. So in that map, we model our protein in that map. And what is not surprising to see cGMP in the non-catalytic side. But there is surprise, and surprise is in the catalytic side. You see, this catalytic site is blocked by gamma subunit, but it has a ligand in that. So what that ligand is, we model that ligand as GMP, but I will explain later. So that's the model. We have ponytail, very similar to ROD-PD, GAP-A, GAP-B domains, and the catalytic domains, and LH2 cross the symmetry axis, such that catalytic domain of one subunit lies just beneath the GAP-B domain of a separate subunit. And the gamma subunit here in green also crosses the symmetry axis. So, and there are ligands in the catalytic side. So in similar fashion, we solve the structure of con-PD with con-P gamma. So the only difference is you can see here the ponytail is missing. We cannot resolve the ponytail. So it's seems to be slightly more dynamic molecule overall. And both of them are highly similar to ROD-PD. RMSDs are fairly small. So what else is common besides overall similarity with ROD-PD? There is conservation of that signal transduction, allosteric communication route between GAP-A domains and catalytic domains that was first identified in Cris structure. So, and you can see this beta-1, beta-2 loops from GAP-A conducting GAP-B, and the other loops are from GAP-B are contacting the catalytic domain. So this is important. So it's conserved in both PDEs. And more importantly, some of these mutations that are found in achromatopsia in patients, they disrupt these communication routes, like those two arginines, 104, 102, that's exactly disrupting GAP-A, GAP-B communication. Another mutation is disrupting GAP-B catalytic domain modification. So the structure actually tells us about some interesting things and links to retinal disease, that R29 W mutation does something to the ponytail. So ponytail is very dynamic, but seems to be important. So anyway, what are the differences? The main differences is the interaction of the gamma subunits, both ROD and con gamma subunits, with the GAP-A domain in con-PD is deficient. We don't see the density going. So in ROD-PD, this is ROD-PD gamma superimposed from the ROD-PD structure. And this is con-P gamma from both structures. Con-P gamma and ROD-P gamma looks in con-PD structure exactly the same. So that interaction is missing, or for the very least is very dynamic. And secondly, as I already mentioned, we see something in the catalytic pocket. And we model that as cyclic GMP, guanidine monophosphate, the product of the reaction. There are two reasons. First, the fit for the density is slightly better, but also we know the enzyme is active. So it must be GMP. And the catalytic pocket is actually very homologous to other known structures of PDEs in terms of interactions, in terms of coordinating metals, which are magnesium and zinc, in terms of guanidine base specificity. So that's another major question is, what is so specific about PD6, which has activity, catalytic activity, at least 2 orders of magnitude higher than any other PDE. But that's for another day, the answer. So I mentioned that we solved first structures with the addition of cyclic AMP or cyclic GMP before plunging grids. But then we've done that without addition, just to check how the PDE without even, we were more interested what happened if there is no ligand in the catalytic domain. But what we see that another subset of subunits appearing that misses gap domains. So it doesn't mean they're not there, they're just flexible and they don't show up in map. And interestingly, most of the particles have both catalytic domains disappearing from the map, but only one small subset of particles from which we were unable to make a reconstruction seems to have a little bit of a density of one catalytic GAP8 domain. So it may be that dissociation of cyclic GMP from one domain destabilizes second enough to cause dissociation or lose density in appearance. And also what we did is also we decided to look, okay, gamma subunit seems to play extremely important role in structural organization. So we trypsinized PDE to remove gamma. And as you can see, the protein is full length, but the full size projections are missing in 2D classes. Only partial projection, somewhat higher resolution features, but they're partial projections. And we were able to map reconstruction match to the GAP-B domains. But GAP-B domains, interestingly, they have different angle where they intersect each other from 29 degrees to 55. So it's like this. Okay, let me see if this morph, yeah, it's showing up. So in morphing, it goes from normal structure to this. And as you can see, dimeric interface with GAP-A domains and the catalytic domains is disrupted. So how is that structure stabilized? So for that, we did molecular dynamic simulation of the model. Started to play and stop. Let me try from here. Anyway, it doesn't matter. So these subunits will jerk for a while, but at the end of molecular dynamic simulation, they would just stabilize, and they stabilize by packing of the catalytic domains to the GAP-B domain. So that's most likely something like that. This is how trypsinized PDE works. But this rearrangement, grammatic, is fully reversible. If you add P gamma back, you get the full projections back. You can reconstruct full normal PDE. So that's how TPDE can be reversed into color PDE almost by adding P gamma back. So that's the summary for the structure. Lack of the interaction with GAP domain for con PDE, or gamma subunits and GAP-A domains. In the absence of added cyclic GMP, significant fraction loses cyclic GMP, which indicates low affinity. And gamma plays critical role in the structural organization. And finally, very quickly, we analyze structural dynamics and using 3D variability analysis. And just to make long story short, this the way you do it, it analyzes variability in a way that we're able to detect subsets of particles that are losing gamma subunit, because we found GMP in the catalytic pocket. That means catalytic pockets, it's getting open, it's floppy. So that's, okay, this is just one component. But look at the other component. Density for the gamma subunit is changing. So we did more extensive analysis by doing DVA and symmetry expanded data sets. And that allows you to quantify all catalytic subunits, individual losing gamma subunit. And as you can see here with this extended data set, that's the only one subunit. It's changing from full density to almost no density. And you can cluster that, and you can remove duplicates from extended data set and do reconstruction. And with all reconstruction, okay, this is reconstruction from data set with rod P gamma. 20% of particles have gamma missing at the catalytic site. With con P gamma, 28% we found. With GAF A less particles, we have 43% of particles losing gamma. So this is a small number of particles that lost both gamma. So what our DVA analysis says that in con-PD there are populations of particles where P gamma density is missing from the catalytic site, but it's still lingering at the GAF domains. And that's likely to be the primary source for spontaneous PD activity. And fraction is increased when P gamma rod is substituted by P gamma con, and when cyclic GMP dissociates from GAF A domain. And I will take just 2 minutes to roughly, is it okay if I take a couple more minutes? So now, if the fraction is increasing when you change rod P gamma substitute by con P gamma, why? And there is one residue here. It's this one residue that is different between rod and con PDE. And that residue turns out to be quite important. So this tyrosine in rod P gamma makes multiple hydrogen bonds, hydroxyl of the tyrosine with carbonyls, backbone carbonyls of a number of residues, large number of theirs. So if you replace it with phenylalanine, then these interactions are going to be lost. And what is interesting is that tyrosine is present early on in fishes and lampreys, and then in close it appears as phenylalanine, and it continues to be tyrosine in the rods. So that's, so we did biochemistry. Okay, we inhibited PDE6 with the rod and con peptides and with the full length. And indeed, we tried to do it carefully. And con peptide inhibit catalytic domain less potently. So that explains flopping, greater flopping of P gamma at the catalytic site. And that explains the higher basal activity of con PDE. Now, and another thing that, I will skip that, except to say, so evolution of gamma subunits happen in 2 steps. One is appearance in rod subunits after first round of whole genome duplication of this domain that I outlined, and that's a GAF A binding domain. That domain appeared in rod gammas to increase affinity of rod gammas to phosphodiesterase catalytic subunits. But then, when rod and con PD genes separated, then cons acquired this F substitution that actually decreases affinity. So rods evolved to increase affinity, and cons subsequently further evolved to decrease affinity, which is, I think, could be important for adaptation to terrestrial vision. And finally, just one word. The GAF domains of con and rod PD are also very, very homologous. If you overlay, it will be hard to tell what's the difference. Why cons have low affinity for cyclic GMP. But of course, the structure suggests that one possibility is that gamma in con doesn't bind to the GAF domain. So the cyclic GMP binding site is less stabilized. But besides that, we found one more reason, and that is FL residue. You see right here, this residue. And F is making pi staking interaction with guanidine ring in the GAF-A domain. And L is not so much. It appears that, and even the opening in the con-PD appears wider because of the L residue. So, and again, it's evolution where L appeared later in the evolution after about amphibians and in the higher species as a way to decrease affinity of con-PD for cyclic GMP. And what we found that actually LF substitution, which we did in the GAF-AB construct of chicken PD. So it's still slow, but you can see for the wild type domain construct, there is different off rate. That this is a cold chase-like experiment. But when you mutate L for F, it's just almost not dissociating. And also we found that CGMP, when it's bound, increases affinity of PD gamma to this chicken construct. So now, just summary. Based on our cryo-M studies, we actually look more carefully at the GAF-A domains and the interaction of P gamma with the catalytic domains and identify these two substitutions, one in gamma and one in the GAF-A domain, that both actually work to decrease affinity. Both substitutions are happening in cons and both reduce affinity of gamma. Cyclic GMP binding affinity GAF domain is allosterically decreasing affinity of gamma, but the YF substitution directly decreases affinity. That these effects to raise basal activity and to speed up con responses. And Vladimir knows much more about how that important to physiology of cons. And I'd like to finish here because there is little time left for questions. I'll be happy to answer.
Thank you, Nick. Let me just start with a few comments. And for those who do not know this field for as long as we both worked in it, this is, I believe, only one protein that cannot be heterologously simply expressed. PDE is such a problematic thing, and still the broad enzyme is not expressible.
Yeah, I completely agree.
And so you have a fellow here who spent his life and solving that problem that end up with the structure. This is really remarkable. And I have to take my hat off to you sticking to this project. Most of the scientists will never stay focused for that long to get this information. And I will just really, I think I said from my heart what I feel. And one more question, if I may. What the heck we have the two subunit in rods and not in cons? Why do we have this heterodimer as catalytic subunit as compared to cons? Is there an answer of some sort?
There must be an answer. I don't know this answer, but we can kind of speculate how that happened. And it's happened probably during the second round of whole genome duplication. But why stay conserved? I don't really know, except there are systems where apparently rod PD subunit can work as a homodimer. There is this chicken genome doesn't have PDA6A, so it seems like at least it's theoretically feasible that rod PD would function as a homodimer, at least in one species. But all I know, it's really clinically very important, because my impression, and correct me if I'm wrong, that PD6 beta gets mutated more often than PD6A.
More severe.
And more severe. So I think actually trying to understand the contribution of which subunit will be the next step that we want. But it's very difficult, and exactly because we still don't have expression system. Is it, we are trying very hard, but so far no luck. Is it because there is some other unknown chaperone? I cannot tell. But what I can tell is there are some reports out there that claim that they can express rod PD. I don't believe it.
Agree. Maybe the only way to do it is to do on mice.
Yeah.
And so hopefully we will get this answer not that long from now.
Yeah.
Hi, Nick. It was a real structural biology and biochemistry tour de force. Thanks for sharing it. One thing I noticed in your gel filtration, the gel showing the gel filtration purification of the PD6C with P gamma, that you still had like a band underneath. It looked like it was SHP 70 or something. It was kind of shifted a little bit. It looked like you had picked fractions.
Oh, yeah.
So my question is, did you look at those fractions where you could still see that, I guess it was SHP 70? Is there some role for this ongoing interaction even after the folding's completed? Do you think that in vivo it's still interacting with these chaperones like the same way that the glucocorticoid receptor does?
Yeah, that's an interesting question, because we do see people, but very little. I mean, it's very little, and it's not stoichiometric. And I think there may be some, even when it's folded, maybe there are some pieces, and like in the structure, you see these loops going from GAV-B to catalytic domains. So these are basically unstructured pieces. And HSP 70 likes to click on those and binds to them. And whether there is some physiological significance to that, but it may be, so there isn't HSP 90 and 70 complement in the outer segment. But maybe when it's folded and starts to traffic, maybe HSP 70 binds to some molecules to facilitate that going towards the cilium. And yeah, because once it's sitting on the membrane, it's it feels good. Feels good. So it's a good question, yeah. Or maybe it's just artifact of our expression system, because we cannot precisely control all viruses. We try to, yeah.
Thank you for sharing this really beautiful work. It's actually inspiring, and I am wondering if there's any evidence of post-translational modifications on PDE6 or its binding partners, such as glycans, that are often overlooked but may help facilitate its functioning.
And post-translational modifications on what molecules?
On PDE6 or its chaperone.
Oh, yeah. This is my fault. But yeah, of course, there is prenylation on catalytic subunits. So con-PDE is geranylated at the C terminal. ROD-PD alpha is farnesylated, beta is geranyl-geranylated. That allows peripheral membrane attachment of the enzyme. And it's very important for phototransduction because in solution, PD is not activated efficiently. The entire activation complex and transducins are granulated on gamma and meristylated or alpha subunits. So it's a membrane complex, and inactivation complex, including RGS9 and R9AP, which is membrane protein, is also membrane. So the whole thing is a membrane bound. And actually the next frontier is studying these complexes. Structural frontier is by cryo-ET or even perhaps by single particle analysis, but using vesicles derived from rods or from retina or even from HEX cells if you express. But then you try, and some people are beginning to do that, try to image these complexes on the surface of vesicles. It's challenging. It's hard to get enough good numbers of particles, but people do it. So that's...
These two mutations that increase the basal activity of COMP-PD6, you think this is because adaptation to light environment? Have you seen in vertebrates that are nocturnal, sometimes it might reverse the mutation, or all the terrestrial vertebrates have these two amino acids?
No, it's, yeah, I'm not, yeah, it's just speculation. And the speculation is because one mutation starts with amphibians, and the second mutation starts with lizards. And this is at about the time where animals move from seas to different to Earth and to different lighting environment. And cones would experience probably much larger high intensity than in other animals. Yeah.
We have a question from the Zoom, Michael Redmond.
Okay.
Hi, Nick. I love your talks. They're always terrific, full of information.
Nice to meet you. Nice meeting you.
Nice
on
meeting
Zoom.
you on Zoom. Yeah. My question is, since you've got the tyrosine to phenylalanine and the phenylalanine to leucine,
Yes.
how are they? It looks like it would be modifying like pi stacking interactions. Can you comment on that? Did I miss it?
Yes. So certainly the Y to L substitution in GAF-A domains, that would disrupt that interaction. And I think that's a part, because that interaction, it's like F2L. That why not F2L? It's that phenylalanine goes just underneath the cyclic GMP that is bound to the GAF domain, and it makes this pi-pi stacking interaction. And it also, it's bulkier than leucine, so the opening into the catalytic pocket, into the GAF-A pocket, seems to be narrow when a phenylalanine is present. And also that phenylalanine is within like about 4 angstrom of the gamma chain, of the gamma piece that interacts with GAF-A domain in ROD-PD. So in ROD-PD, this combination of gamma and phenylalanine would solidify, I mean, very restricted opening of the and in fact, my personal view of the role of GAF-A domains and non-catalytic cyclic GMP in ROD-PD is not regulatory. I think it's more structural, because I don't think anybody's seen full-length rod PD without cGMP bound to the non-catalytic side. In con-PD, that's a different story, and we want to understand what exact physiological role for that. But we know that con-PD can lose that cyclic GMP, and losing that cyclic GMP dramatically affects p-gamma affinity, overall affinity for the enzyme. So, but you're right, exactly, that's what's happening. Oh, and just quick thought, it's that why in the catalytic pocket, why is making contact to the backbone carbonyl? But it's one of those residues, it's phenylalanine. And it's actually making pi-pi staking interaction with GMP in the pocket. So, but I have no idea what the significance that could be. But it may affect theoretically release of GMP from the pocket.
Okay. Thanks. Terrific.
Hi. In your expression system to study maturation of con-PD, you say you add PD gamma, and that leads to the appearance of the full activity. Was it con gamma? Was it rod gamma? Or
both?
They
both.
Could be interchangeable.
We use both, but in the original study, I think, yeah. So we use both. Both have the on maturation. They have the same effect. So it's probably expressing the level of expression of gammas exceeds KIs. So basically, or exceeds the affinity is higher than the affinity cost. So that means they all bind, and maturation is complete with gamma. Oh, one interesting note, but I don't know why. In hex cells, addition of gamma has 100-fold elevation to get full activity. In SF9 cells, we see 10-fold. 10% of PD can fold without in SF9 cells, but I have no explanation for that. It's just different cell systems.
Hello, very interesting talk. I have just a question regarding maturation process. What is the role of PT motif in this? What would you suggest, if there is any?
PT motif in maturation, I think it could initiate dimerization. So it's like point initiation of dimerization. How and because it's so flexible, I don't know, but that could be the role. So, and like mutations in PT motif linked to disease do say that it's important. Yeah.
One follow-up. Are there any specific domains that are more important for this maturation process in PD6? When they are mutated, it will disrupt more, and some of them are less important.
I think for maturation is the catalytic domain is the most important, I think, but it's not because it's unfolded before the maturation process, but I think it's improperly oriented, or I mean, there is even chance that like in PD2 that the catalytic domain actually gets the side catalytic is occluded between the two catalytic domains, and it's clearly, well, we call it misassembled. And what we know is once it's misassembled, then APOL or p-gamma would not bring it back. You have to act before it's completely locked in the misassembled state.
Hi, Dr. Artemyev. Thank you so much for an interesting talk. So my question is about the maturation of PD. So APO deficiency causes LCA type 4. So is it, this might be a naive question, but is the severity of the phenotype a direct loss of the protein itself, but also is it also a complication due to the loss of this obligate chaperone for the PD6 protein?
I think it's because PD gets lost earlier than with mutations. Mutations, they can still fold. So some mutations may be folded perfectly, and they traffic to the outer segment, but they, let's say, catalytically inactive. But they have additional functions. For example, Cree's studies suggest that they may PD can act as a linker between two disks. So it may have structural role or something else. Or let's say before PD is completely gone, one subunit is partially active. So that slows degeneration. With LCA, if they misassemble both rod and cone PD, there is nothing. So that means both rods and cones degenerate simultaneously and very severely and very early in life. And yeah.
Hi, thank you so much for your wonderful talk. I have just a curiosity. Have you checked the level of PD6C protein and activity in RD1 mouse model? So without PD A and B?
In RD?
In RD1 mouse model?
Yeah, RD1, actually, in RD1, yeah, we have, when I told you that there is no maturation of PD in the absence of P gamma in rods, is we know that there is little or no activity. But again, there is con PD activity. So to really rule out if there is any PD activity, you have to do, we did double knockout. We took, we use actually different mouse. We use P gamma knockout, rod P gamma knockout mouse. And so it's not RD, but it's somewhat related to equation. And we bred this mouse to CPFL1. And that is con knockout. It's con PD is lacking. So, and then we looked for PD activity in that mouse after everything degenerated. And in that mouse, oh yeah, and in that mouse, there was no PD activity above background. So like if you take P30, everything degenerated in the double knockout mouse. And you still can measure like 0.01% activity. But that activity is the same P10 and P30, meaning this is not PD6 activity. So, but in that study, if you look in that study, I may not give you exact number, but I think it's a small fraction. Con PD activity, we estimate at 2 or 3% of PD activity. Yes.
Hi, thank you for a wonderful talk. My question is about the mutations that disrupt the communications between the GAF-A domain and the cavity domain. Since the affinity is reduced in cones for the GAF-A, or in cones in GAF-A for cyclic GMP, I'm wondering if the effect you see in catalysis is more drastic in cones versus rods for those mutations that are disrupting allostery.
Oh, we have not studied them directly. These are mutations in patients. So I can imagine two mechanisms, but we have not studied them. One mechanism would be effect on maturation, because mature protein is stable. So you want, so they may affect maturation by making protein less stable. But secondly, when and if the enzyme with such mutation matures, then I would say I would still think that mutant would be at least catalytically active. But I think those mutations would affect probably gamma binding and effects of non-catalytic cyclic GNP on the catalytic activity. So you would see some predicted phenotypes. But I think before those conclusions, you want to know what those mutations can do. Because it's remarkable. So there is related, structurally very similar PDE5, very important in pharmacology and for various drugs, and is used already to treat multiple diseases. And why do you think there is no cryo-EM structure of full-length PDE5? And I think, just my opinion, there is no full-length structure because there is no stabilizing elements to that structure, as in PDE6. And this includes those allostatic pathways, one element. Second element is P-gamma.
Hey, Nick, beautiful work. And it's great to see that your structural analysis showing the lower binding affinity of COMPD actually seems to explain the higher spontaneous activity of COMPD that has been measured. I've also really always wondered about why rods have two PDEs and cones have one, and it sounds like Chris asked the question. We still don't have the answer. So I'll ask you, is there any, because you've done evolutionary analysis, are there any differences between mammalian and non-mammalian PDEs that might seem to adapt to the higher body temperature of mammals?
From just sequence analysis, it's really hard to tell because they're highly homologous. So what we know, all we know is from biochemical studies, and there aren't very many. We primarily use mouse and human as a recombinant. So there is some early work on frog PDE, and in that, those early studies would suggest that transducin activates frog PDE somewhat easier, and even like transducin P-gamma can dissociate in frog PDE. So that means that potentially gamma is bound less tightly. But it's not because of the YF substitution, because in frogs it's already, rod PD, yeah, of course, would be Y. So it's not F in frog, in OPD. So, but there are biochemical differences. We probably need to look very carefully at frog PD, and maybe I think frog PD is doable to do by cryo-EM. The question is it now so critical to do frog PD? We all worry about human health. Yeah,
okay, thank you so much.
There are some.
Nick, this was really wonderful. And again, two lessons from today. One is we learned quite a lot about PDE, but the second, you set the important problem and then you solve it, regardless what it takes. And with one more note, Mike Redmond, are you there? Mike is not there. I think he just left, but Jim is there. Jim, just remember to buy flowers. Thank you.
Thank you very much.